Thank you. Yes. So, right. Now it's just showing me this meeting is being recorded, but maybe that's straight. Okay, so my plan for today is to talk about, the title, right? Machine learning and how you can apply it to study somatic evolution in cancer. But I sort of have targeted this for computer scientists that don't know too much about biology. And I know that some of you know biology, so it's sort of perhaps not perfect, but still. So I'm going to talk a bit about evolution, somatic evolution, not too deeply, the kind of data we work with, and a bit of machine learning methods that we apply. And if you want to interrupt me and ask questions, that's excellent from my point of view. Yes. Okay. Right. And as you heard, I'm at SciLife, and I also belong to CSD at the ECS. Okay. Now, can I? Yes. Okay. So just start with the sort of general methodology that we apply, which is not. I mean, it has several mathematical components, but it's easy to communicate. So we typically formulate some sort of probabilistic model as a probabilistic graphical model. So, and there's then observable variables which corresponds to things we can see in data. And then there's a number of latent variables which correspond to what we want to know. And then we typically want to infer a posterior distribution over those latent variables. We may sometimes work with a maximum likelihood approach also, but typically we do it in a Bayesian way. So we're after the posterior. And we use tools like MCMC and sequential Monte Carlo, Gibbs sampling, etc. And those of you who are familiar with those techniques know that, well, actually it's not one or the other, but you can mix them and sort of use them together. There's things like particle Gibbs, etc. And these techniques are nice because we're interested in evolution, for instance. But when you're interested in evolution, you tend to study trees. And these techniques can be used quite easily to study trees or infer a posterior distribution over trees. We also like these techniques, variational inference and also the variational autoencoder, which both are, in particular, the variational autoencoder is a little bit more new. And we like those because they're fast and many of our data sets are large and we want to be able to analyze large data sets. A disadvantage with these are, well, it's not so easy to infer posterior over trees using. Okay, now, right. So when I was a kid and I studied biology in school, I didn't find it interesting at all. I just sort of saw it as a collection of facts, unrelated, etc. And but today I know of this paper. It's a bit worn. I wouldn't have used this in a normal presentation, but it's a paper by Dobzhansky, and it's sort of in general about evolution, and the title is "Nothing in Biology Makes Sense Except in the Light of Evolution." And that's also true for cancer, as we will see soon. Okay, but today we can see many studies of evolution. This is an example of an evolutionary tree of a number of virus samples. In contrast to this tree, where these leaves are basically sampled at the same time, here the viruses are sampled at different times, so the leaves appear at different time points. But the purpose of studying a biology like this is typically, well, there's some interesting strains, and you want to know how they relate. In this case, there's a number of so-called mutants, which should actually be called evolved strains, that are of particular interest, and the researchers wanted to know how they related. This tree was constructed using a software called Beast, and which is a Bayesian package for analyzing evolutionary trees. And I just want to point out the huge difference between constructing a package like this and using it to construct the tree. Right? Okay. There are other examples, sort of much more deep examples of how you can use phylogenies. And this example is from actually researcher that you can find at SciLife, not in Stockholm, but in Uppsala, Siv Andersson, who 20 years or so ago sequenced rickettsia and found out that when you construct an evolutionary tree, rickettsia is sort of placed close to mitochondria. And the conclusion from that is that actually rickettsia, which is an alpha proteobacteria, is closely related to mitochondria in eukaryotic cells. Actually, the mitochondria comes from an alpha proteobacteria. Okay, so there's lots of applications of evolution, and in particular, construction of evolutionary trees. And if you today would say, oh, you know, look at sort of the amazing biodiversity around us, and isn't it a miracle that it exists? Then people typically would say, no, it's not a miracle. It's pretty well understood how it, you know, has come into place. It has evolved, and it was figured out by Darwin, right? That evolution is random mutations and selection. And that's exactly what's sort of behind this scenario and also this scenario, right? It's of course not the case that it explains exactly how things happen, but it explains the basic rules, random mutations and selection. But when you talk about cancer, when newspapers write about cancer, it's often portrayed as a mystery. You know, it's like, oh, how can this exist? But if we recall the words of Dobzhansky, nothing in biology makes sense except in the light of evolution. So we should probably try to find an evolutionary explanation of cancer. And now I'm not talking about how sort of humans have evolved and obtained cancer in relation to humans, but how actually a tumor progresses, is created and progresses. Right? So, and this was figured out, say, in the 70s by people like Peter Noble. And the story goes as follows. And that's depicted here, right? So there's some healthy tissue illustrated by yellow here. And there's one cell that gets a mutation, which makes it proliferate more than the other cells. And merely that increases the probability. So that's the one that gets a mutation, right? That increases the probability that one of its descendants will get the second mutation, these two. And you know, this will go on and go on. So, and result in a tumor, which typically has a large number, or often have a large number of subpopulation or subclone, as they're called. And these subclones can then have widely different phenotypes. You know, they can have widely different properties. And normally when we talk about wild species, we like that species to have a lot of genomic diversity, because genomic diversity is a strength. If a lack of genomic diversity can easily result in, well, you know, the extinction of the species because of a virus or whatever. And it's the same thing here. You know, genomic diversity in a tumor is a strength. but it's a strength for the tumor, not for the person carrying it. And for instance, it may be the case that this subclone is resistant to a certain therapy while all the other ones are not, right? So diversity gives strength. As usual. Okay, but what are the types of mutations that we're talking about here? Well, it's copy number alterations and also point mutations. So point mutations, I want you to think about the normal genome, like, well, there's 2 copies of each chromosome in general. And we typically have homozygous positions. So both these copies have the same value in any position, A, C, G, or T. And then a point mutation is simply one of these. In this case, this one is altered and becomes another nucleotide. Right? So that's a point mutation, it's a nucleotide substitution. But then there's also copy number alterations. So again, normal genome, but a cancer genome can have different number of copies at any position of the genome. So you can have 2, like the healthy, 0, 3, 4, etc. And then these can of course be mixed also. So a genome can carry both copy number alterations and point mutations. But I will focus on the point mutations and sort of assume that also the cancer genome has copy number 2, like normally, just to simplify things. Okay, so today there's a number of interesting experimental techniques that we can apply to a tumor and other tissue. And 2 of them are, well, we can take a single cell from a tumor and sequence its DNA. And I'll give you a simplified view of what sequencing is in a moment. We can also take a cell from a tumor and sequence its RNA. Okay, so DNA, well, we get to see the mutations in the DNA. RNA, we get to see the activity levels of the genes, but we can also see mutations in those genes. Okay. Then we can also apply spatial techniques, but I won't talk about those today. So one thing we can do using these techniques is what we can sample single cells from a tumor, and then based on the mutations they carry, we can reconstruct the tree that led to them using techniques similar to those that I teased, etc. So this can be done on the single cell level, but it can also be done on a clone level. So then the aim is to reconstruct clones, basically these colored clones here, right? And that's what I will be talking about today, mainly. Okay, what about sequencing? It's a complex technology, right? But from a computer science point of view, you can see it more like an urn model, okay? So there's something called bulk sequencing. It's when you take DNA from typically DNA, well, you can take DNA or RNA, but here I will only be talking about DNA. So you take DNA from a tumor and you sequence it, which basically means that time after time you reach into the tumor, you pick a piece of DNA of length, say 300 or so, and you get to see its sequence. Okay. You can also map this to the healthy genome in order to see where it came from, and you can then also see if it's mutated, etc. That's called mapping, and it's sort of figured out how to do. So as soon as I get the piece of DNA, I can map it to the genome. Now, what I cannot do when I do bulk sequencing, or pick from this urn, I cannot see whether 2 pieces of DNA that I pick up come from the same cell or not. But if you do single cell sequence, you can do that. Because then, in that case, if you do single cell DNA sequencing, you only have one cell in the urn and you pick from its DNA repeatedly. Okay, or you do single cell RNA sequencing. You have a single cell in the urn and you pick repeatedly from the expressed genes, sequences of length 300 or so. And of course, those genes that are highly expressed, you have a large chance to pick a sequence from those, and those that are not so highly expressed or not at all, well, then the chances are smaller. Okay. So the interesting thing with single cell is exactly that when you do bulk sequencing, you don't know if you pick DNA from the same cell or not, or RNA from the cell. But if you do single cell, it's like you have the cells in different urns. So you pick from this, right? And as long as you pick from this, you get DNA or RNA from that particular cell, and then you go to the next one. And that's what we will be using. Okay. Now, so yes. Now, say we have a tumor. We want to figure out sort of subclones is there in the tumor. So is it only one, or there multiple, etc.? Does it have genomic diversity or not? And I'll run this example where we try to figure that out based on first bulk DNA sequencing data. And now we sort of start to use single cells. Okay, and I will, in the example, consider two different possible tumors, one with this clonal tree and one with this clonal tree. In both these examples, well, what we have here is the figures on the vertices of the tree is the frequency of that subtree. Okay, so half here means that half of the cells belong to this subclone or one below it. And it's the same thing here. Half of the cells belongs to this subclone or one below it. Okay, and but there's a huge difference here because, well, below this one, there's a subclone which has a quarter of the cells, which means that the cells that live exactly at this clone is, well, it's a quarter of them, right? While in this example, well, below here, there's half of the cells, but then you have a quarter there and an eighth there, which means that all the ones that live exactly there, that's an eighth of the cells in the tumor. Okay, so don't worry too much about that. The important thing is you look at one level here, and the number tell you what fraction of the cells belongs to that subclone or one below. Yes, okay. And as I said before, I assume that the copy number also of the tumor cells, the cancer cells, is 2. And that makes it easy to understand then, if we have mutation, or as I will be calling them, single nucleotide variation, SNV, think about it as mutation. We have one introduced here. Well, then all the cancer cells are carrying that mutation. So if I now reach into this urn and I pick up a piece of DNA from the covering a certain SNV, then, well, half of the chromosomes carry the mutation. So we expect to see half of the reads having the reference, that is the same thing as the healthy genome, and half of the cells having the alternative, right, which is the mutation. And this is the same thing if you go down. So, if I look at the cells, the reads that at a certain position carrying an SNV introduced at this level, well, half of the cells have that SNV. Okay, they only have it on one of the chromosomes, and the other half of the cells don't have it on either of the two chromosomes, which means that yes, the alternative will be a quarter and the reference. And this just goes on then. So if we look at the SNVs introduced here, well, that's a quarter of the cells, and 8 of the reads covering that SNV position, that position will be alternative mutated, and the rest will be, the 8 will be alternative, and the rest will be reference as the and the same thing for this guy. So it's just, the alternative reads are just half of the fractions you see in the tree. So that's nice, but we were interested in this clonal tree, which I'll be calling the cherry, and also the path. Okay? And it's the very same thing in the path. If you look at an SNV introduced at this vertex, well, an 1/8 of the reads will carry that. An 1/8 of the reads covering that position will carry the mutation. And the same thing over here, it's half of it, which means that, it's the very same thing. There's no difference between the cherry and the path. But based on bulk data, it's hard to distinguish these two cases, and they may be, clinically relevant. So that's where the single cells comes in, because if you look at a single cell, this one is supposed to be a cell that belongs to this subpopulation. Okay, and we look at its genome, and there are 2 positions. One in which a mutation is introduced in this subclone, and one where it's introduced in this subclone. Okay, now, so if you have a cell from here, it will carry both mutations. Okay. The ratios are what the ratios are, as we looked at before. Now, in contrast, if you have a cell from this population, which based on bulk data is hard to distinguish from this, in this case. If you have a cell here in the cherry, well, it will not carry any of the mutations that's introduced here, right? Because it's not the descendant of that. Which means that, it will only have this mutation if the other one is introduced in this. So that's how we can distinguish between the cherry and the path. For instance, for this vertex, they behave in the same way. 2 mutations, one introduced there, one introduced there. Cells from the yellow only contain the one introduced at the yellow, and the same thing here. But for this, it's different. Okay, let me tell you a little bit about Python, which is a program for doing this type of analysis. And this is the complex graphical model that they describe in the paper. And let's simplify it a bit. We will still have this thing, which is the Dirichlet process. It's also called the Chinese restaurant process or the stick breaking process. And I will describe it to you soon. Yes. So let me do that exactly now, actually. So, and let's pick the Chinese restaurant version of it. So there's 2 interesting parameters. One is alpha and one is age, which I will return to soon. Okay, and the process works as follows. There's an anonymous data point that shows up and ask the head waiter to sort of become a real data point. That is a point on the x-axis in this case. It could be something more complex, but in this case, we have distributions that give values that are points on the x-axis. And then the head waiter will do 2 things. First, it will assign the data point. We will assign the data point to one of these tables that we have here. And then, depending on the distribution present at the table, the data point will actually get the value, right? So if the data point is placed at this table, it will typically be a smaller value than if it's placed on this table. And that's also smaller than if it's placed on this table, right? So, okay, so the scheme is the anonymous data point shows up, the head waiter placed the anonymous data point at the table, and the data point gets an actual value from the distribution present at the table. And let's break that into steps since, well, that's a natural way of doing it. First, we have the placement procedure, and that's done based on the number of people seated already at the tables. In this case, we have N1 people here, N2 people there, and N3 people there. And then there's also the possibility of opening up a new table. And we can say that we have a pseudo count of a number of people here. There's nobody sitting at the table, but it's like we pretend that there's alpha people sitting at this new table. And then the placement is according to the principle, popular tables get more people, or the rich get richer, right? So the person is placed here with this probability. The number of people sitting at the table divided by the total number of people plus the pseudo count. And then the same thing for each of the tables. And in particular here, the probability of opening the new table is the pseudo count divided by the total number of seated customers plus the pseudo count. Okay. Yes, and in our case, the data point, well, it's pretty anonymous, but it has sort of a total count. Or it is the total number of reads mapped to this position in the genome. And the question is, the data that we're going to fill it with is the number of mutated reads or alternative reads that we see from the position. And yes, the placement is exactly as before. And then, if the data point gets seated here, its value is picked according to a binomial distribution with parameter total number of reads plus this phi parameter, right? And the phi parameter is decided when new table is open. So when the new table is open, the phi value is sampled from a beta distribution, which means that the distributions we have at the tables are beta binomial distributions. Yes. Okay, and what can you do with this? Well, there's lots of things, exactly like phylogeny in general, the examples I started with. Here are two examples. One is where these guys looked at tumors. They divided them into those that only had one subclone, where they appeared in a chain, like that, and where it was a branching structure, so the cherry or a more complex tree like this, right? And for these cases, they looked at the number of tumor infiltrating lymphocytes, so the number of immune cells present in the tumor. And they noticed that for the pure ones, there are more infiltrating lymphocytes. The change, well, less. And for the branched ones, even fewer. Here's another example where they looked at loss of HLA. HLA is involved in the mechanism that holds up peptides on the outside of a cell so that the immune cells can see whether the cell is carrying a virus or perhaps a mutated genome, a cancer genome. Right? And all of this is, well, you could say, well, yeah, but I mean, the immune system is working like it does, and let's just let it work. But that's not what's going on today. Today, there are therapies where you can stimulate the immune system. So it's extremely important to understand which patients would have that therapy, where is it functional, and how does it work? Okay, so let me talk a bit about a software that we constructed, which is doing, we call it phylophenotypic analysis. It's constructing a clonal tree, and for each of the clones, it's also figuring out sort of the phenotype, how it behaves, right? What type of clone is it? That is the expression profiles for the genes. The expression profiles for the clone. So in a clone, which genes are expressed and which are not expressed. And we do this based on expression data, single cell expression data. We use, how am I doing in terms of time? Should I sort of try to?
I think maybe another 10 minutes or so.
Okay.
Should be fine.
Sure. Yeah. Okay. Yes. Great. So in order to do this, we use another prior than the Chinese restaurant prior, and we use sort of the tree version of it, and it's usually called the tree-structured stick breaking process. It's using the word stick breaking, which is an alternative name for the Chinese restaurant and Dirichlet process, the stick breaking process. And in this case, there's a tree, and you seat people at vertices. So basically, the tables are connected in the tree structure, right? There's a room, and the following scheme is used to seat the new customer. First of all, the number of customers already seated at the vertex is NV, for vertex V, and the number below, not including V, is N less than V, okay? This thing here. And now there's a new customer showing up. How do we seat it? Well, what is the head way to do? Well, it starts to consider a root and then sort of traverse the tree downwards, right? When at the vertex, you seat the customer with this probability. The number already seated at the table, +1, divided by the number of customers seated at the table or below, plus, well, this is sort of pseudo count, right? +1. So the alpha is the parameter. And if you decide not to seat the customer at the table, you have to decide which child to continue with. And you pick a certain child C of the vertex you consider with respect to the number of customers seated at the vertex C or below. And you divide by the total number of customers sort of seated here, right? That's V, that's C, and you divide by the total number seated below V. That's gamma. Again, a pseudo count. Now, opening up a new table is the same as creating a new child, and you do that with probability gamma divided by the total number of guests seated below V. So it's sort of a cute little way of seating people, which gives a prior distribution, the TSSB. We use it. We use the beta binomial distribution, exactly as I described earlier. We use Monte Carlo Markov chain MCMs to sample. It's a bit of slight sampling, etc. So these are concerned with the actual model. This is the sampling in order to infer the posterior. And as many of you know, when doing MCMC, what you actually do is that you're at the state, you propose a new state. In order to know whether you should move to the new state or not, you need to be able to compute the likelihood of the states, or the joint probability, the prior and the likelihood. So that's what we need to do. And the key in our case is to say, well, I have this cell here, or rather the single cell expression data from a cell, and my state is some sort of clonal tree, like this one, the cherry or the path. And now I want to say, well, you know, how well the clonal tree is describing the data has to do with how well this cell fits the clonal tree, which may look like this or this, okay? So yeah, let's say that we're considering two S and V positions. We have the total number of reads and we have the number of reads with which have the alternative mutation. Then it seems to be good to use this rule. So if you have a clone with a mutation at the position I, all the alternatives should basically be like half of those of the total number, right? So you should, AI should behave like if it's sampled from this binomial distribution. And if you don't have the mutation in the clone, well, then it should be not a half, but some almost zero probability of getting the alternative. So epsilon, a smaller value epsilon. Best. But unfortunately, or fortunately, if you like modeling, biology is a bit more complex. And for that reason, what we use is not just this path, but we use a mixture between two binomial distributions, which both have priors, which are beta distributions. And in one case, we basically have what we had before, like a half, which is a beta distribution like this. And in the other case, we say, well, it's actually like this, that cells sometimes only express one allele. So you may see the reference only, or you may see the alternative only. So in this case, in order to model that, we sample this phi from a beta distribution that looks like this. So it's either phi is either very close to 0 or very close to 1. And otherwise, well, then we basically behave like before. Okay, and does this work? Yes, it does. So if we create synthetic data from this cherry, and then we run our program, which is called Phylex, then we get the right thing. While if we use only bulk data, you know, we don't know if two reads come from the same cell or not, then yeah, the best programs give us something like this. Okay. Well, I don't want to bore you with too much of verification of the program, but you know, in general, when we compare with bulk, this is bulk, and we're up here. This is the V measure, which is a measure for how well we cluster the SMVs. We do really well, and not only that, competing processes which actually are aimed at analyzing DNA data, but they can still be used in our case, they don't improve when they get to see more cells, but we improve very clearly when we get to see more cells. We also use this for some biological data where we basically know the truth, because these are some cancer cell lines that have been assayed very carefully in this paper. And so this is ground truth according to this, let's say, let's call it careful, expensive, and reliable investigation. And this is what we get. We get almost the same thing. We happen to collapse a bit where it's actually very distinguished between two clones because one of them has only one S. Okay, and when we use other tools to do this, they don't get a very good V measure, but we do. Okay, now the goal is typically not to get only the clonal tree, but also to get the phenotypes, right? So and the same guys as did this careful analysis, they also devised the method to take, because the careful analysis is based on DNA data. So they devised a method to take RNA data and map onto the clones in order to see which genes are active and which ones are not. So you can sort of see the phenotype of the clones. And we get that for free because we work with RNA data directly. So we get their expression profiles directly. And you can ask then, how well is this working? Well, this is an example where we analyzed HER2 positive breast cancer tumor. It's from Johan Hartman, a collaborator. And we got this actually path in this case. So we have clone 1 up to clone 8. And then we looked at the genes expressed in this clone versus the genes expressed in this clone. And the ones that are more highly expressed in clone 2 are over here. And first of all, this ERBB2, that's a well-known gene associated with this type of breast cancer. And all of the other ones are also sort of typical cancer genes that one could expect to be expressed during the progression of a cancer tumor. So that seems to work very well. Yes, and well, let me just end by first thanking our sponsors, which is SSF, VR, but also EU. And then thank you for the attention.